Hi, I'm Carla Angelica Pasmay-Cotto. I'm going to talk about therapeutic clearance of amyloids by antibodies to serum amyloid peak component. Definition. Amyloid is an abnormal protein that is produced in your bone marrow and can be deposited in any tissue or organ, causing a disease called amyloidosis that can commonly affect the heart, kidneys, liver, spleen, nervous system, and digestive tract. The specific cause of your condition depends on the type of amyloidosis you have. The neuropathogenesis of Alzheimer's disease is believed to be triggered by the accumulation of the toxic amyloid beta protein in the central nervous system. There is no cure for amyloidosis, and severe amyloidosis can lead to life-threatening organ failure, but treatments can help you manage your symptoms and limit the production of amyloid protein.
Interact with the surface of our nerve cells and then cause the problem. So amyloid is a sticky protein that has this tendency to clump together, and it's produced from a large precursor protein, the amyloid precursor protein. We're trying to understand how it's produced, how it then clumps together, and how those clumps of protein interact with the surface of our nerve cells and then cause the problems that we see in people who have Alzheimer's disease. If we could understand how the amyloid is produced, how it's clumping together, how it's interacting with the nerve cells, then there's the potential there to disrupt those processes as potential treatments for the disease. And so a lot of what we do is just trying to understand the basic processes that are going on in the brain and whether we can then exploit any of those as a potential therapeutic target for the future development of drugs.
Amyloidosis is the term used to describe a group of diseases in which deposits of an abnormal protein called amyloid form in tissues and organs throughout the body. Proteins are important molecules involved in almost every structure and function within our cells. They are made up of chains of smaller units called amino acids, which fold themselves into three-dimensional structures. Folding allows the protein to work properly and interact with other molecules. In amyloidosis, proteins are made in the normal way, but when they are released from the cell, they open up again. These abnormally folded proteins collect together in long chains or fibrils. The deposits they form are called amyloid. Amyloid fibrils are difficult to break down, and as they accumulate, they start to cause damage. Eventually, the build-up of amyloid fibrils stops the affected tissue from working properly. AL amyloidosis is caused by a bone marrow disorder. The bone marrow produces blood cells, including plasma cells, a type of white blood cell that forms part of the immune system. Plasma cells make antibodies to help fight infection. Each antibody is made up of 4 protein chains, 2 heavy chains and 2 light chains, which attach to each other to form the complete antibody. In AL amyloidosis, the abnormal protein that builds up and causes problems is the antibody light chain, hence the name AL amyloidosis. Instead of attaching to heavy chains, the abnormal light chains open up and bind to other abnormal light chains to form amyloid fibrils. These gradually build up over time. Amyloid light chain fibrils can occur almost anywhere in the body, but the organs most commonly affected include the kidneys, the heart, liver, spleen, nerves, and the digestive system. Treatment for AL amyloidosis involves chemotherapy, steroids, high-dose therapy, and stem cell transplant, and other drugs. Treatment is aimed at reducing the plasma cells to stop further amyloid light chains being made that would otherwise build up in tissues and organs. It also gives the body a chance to break down existing deposits naturally. However, on its own, this process can take many months or years. New treatments which break down the amyloid deposits more quickly are currently being tested in AL amyloidosis. The hope is that they can reduce symptoms and restore function quickly, and in doing so, improve quality of life for patients.
So what is amyloid composed of? So it's typically altered calcitonin secreted by the tumor cells because of, because of C cell hyperplasia. That's the key thing that's responsible. If you see C cell hyperplasia on the exam, go with this. And this is going to help you differentiate from other diseases. You can see over here all these cells are increasing as well.
Applications in medicine. The amyloid fibril deposits that cause systemic amyloidosis always contains a non-fibrillar normal plasma protein, serum amyloid P component, or ASAP. The drug CPHPC efficiently depletes ASAP from the plasma but leaves some ASAP in amyloid deposits that can be specifically targeted by therapeutic IgG anti-SAP antibodies. In murine amyloid type amyloidosis, the binding of these antibodies to the residual SAP in amyloid deposits activates complement and triggers the rapid clearance of amyloid by macrophage-derived multinucleated giant cells.
For many years, AL amyloidosis was an orphan disease. We were trying to get drugs from myeloma treatments, but now there are effective treatments. We are able to control the disease, and a lot of new drugs are being tested in amyloidosis. The treatment in amyloidosis is moving forward in two different ways. One is we've got new types of drugs which will kill the abnormal cells in the bone marrow, similar to the standard treatments, but they're improvement on the standard drugs. So we've got two new proteasome inhibitors, or Velcade-like drugs, one of them called as carfilzomib and another called as ixazomib, which are both highly effective proteasome inhibitors. Carfilzomib is given intravenously, whereas ixazomib has got the advantage of being taken as a tablet every week. Both of these drugs are being studied in amyloidosis. We are running clinical trials with both these drugs to assess their effectiveness in amyloidosis. There is a new monoclonal antibody which is very effective in killing plasma cells called daratumumab. This is a drug which has been extensively studied and appears to show great promise in myeloma treatment, and a clinical trial for this is being planned for amyloidosis, hopefully starting quite soon. And we think this will be a relatively non-toxic but highly effective treatment in patients with amyloidosis, particularly those with heart involvement. The key issue in amyloidosis is not just killing the plasma cells and reducing the light chains, but to improve the organ function, to actually clear the amyloid protein deposits. For many years, we had no means of actually doing this. We know that in the body, there are immune cells or scavenger cells which will eat away the amyloid deposits. There are new immunotherapies which can accelerate the clearance of amyloid deposits from the body. And there are 2 or 3 such antibody treatments which are being studied in amyloidosis. One is an antibody developed by a company called Prothena, called NEO-001. There are 2 phase 3, which means very advanced phase clinical trials going on. One for patients with amyloid in the heart in the newly diagnosed setting, and one for amyloidosis in patients who've already got heart dysfunction. And we hope that this antibody will go to licensing provided these trials show good results. There is another approach that we've developed within the Amyloidosis Center in the UK, targeting a small protein called SAP, the same protein that is used for SAP scanning for imaging amyloid deposits. This is a protein which is present on all amyloid deposits, and we have a monoclonal antibody which can target these deposits. We have done a small early phase trial with this antibody which has shown very rapid clearance of protein deposits from the internal organs, and hopefully this antibody will move forward into future trials in the future. There are
Pros and cons. Pros. Reduction of amyloid-based disease and major compression over associated disease such as Alzheimer. Cons. The treatment is effective in visceral amyloid deposit, but not in heavily amyloidotic organs, therefore neither in massive amyloid loads.
In this video, we will cover the signaling events involved in the processing of amyloid precursor protein, commonly called APP, that leads to the formation of amyloid plaque. In a normal brain, an enzyme called alpha secretase acts on APP and cleaves it into secreted APP alpha, also called SAPP alpha, and an 83 amino acid long membrane-bound C-terminal fragment called CTF83. Alternatively, in an Alzheimer's brain, an enzyme called beta secretase acts on APP and cleaves it into secreted APP beta, also called SAPP beta, and a 99 amino acid long membrane-bound C-terminal fragment called CTF99. In normal signaling, CTF83 is further cleaved by a gamma secretase complex made up of PSEN1, PEN2, APH1, GSAP, and NCT. Cleavage of CTF83 leads to the generation of APP intracellular domain, otherwise known as AICD fragment. AICD fragment then translocates to the nucleus, where it affects the transcriptional regulation of several proteins and drives neuroprotective pathways. It's also important to note that SAPP alpha gets secreted from the neurons and drives normal synaptic signaling, leading to synaptic plasticity, learning, memory, neuronal survival, and emotional behaviors. In Alzheimer's disease, gamma secretase complex again is assembled, but instead cleaves the CTF99 fragment into an AICD fragment and an A-beta 4042 peptide. AICD is again translocated to the nucleus, where it affects the transcriptional regulation of several proteins and drives neuroprotective pathways. The A-beta 4042 peptide, however, is involved in several downstream pathways related to Alzheimer's disease. A-beta 4042 initially interacts with APOE, which results in aggregation of A-beta oligomers to generate amyloid plaque. Amyloid plaque can be detected via immunohistochemistry staining using A-beta 42 specific antibodies such as clone MOAB2. Downstream, A-beta 4042 with APOE also interacts with MGLUR5, NMDAR, and alpha-7 NACHR, as well as leads to reactive oxygen species and oxidative damage. All of this results in blocked ion channels, disrupted calcium ion homeostasis, dysregulated energy glucose metabolism, mitochondrial oxidative stress, and neuronal apoptosis, which ultimately results in dementia or memory loss.
Issue applied in Ecuador. Amyloidosis is a rare disease affecting around 3 to 5 people per million every year, and that's not different in Ecuador. For example, in the statistic of hospital discharge 2018 by the INEC, 26 people was described with amyloidosis, or in a study of ACPL, cutaneous amyloidosis, in the hospital Luis Bernaza, out of 7,200 people, 195 has ACPL. So we can conclude that the disease is present but have a very low prevalence. Conclusion. Amyloidosis is a rare disease affecting around 3 to 5 people per million, caused by the accumulation of an abnormal protein called amyloid, and causing damage in many organs and systems, being related to chronic disease like Alzheimer, but this is being studied and multiple treatments are emerging as the use of antibodies plus CPHPC showing great results. Thank you.